My name is Roy Cheris, and I have been involved in manufacturing, quality assurance, in sterile and aseptic processing, microbiology, microscopy, and inspection for over 40 years. I am the founder of Bridge Associates International Consulting and Inquest Science Corporation that provides training and guidance on particulate matter and physical defect control, optimized inspection, along with expert defect lifecycle monitoring systems. I'm a member of the USP Expert Panel for Visual Inspection, and I've served on many PDA task force. The title of my talk today is Quality Metrics and Data Integrity Considerations for the VI, Visual Inspection, Particulate, and Physical Defects Lifecycle in the 21st century. And I emphasize that 21st century because our data integrity and quality metrics deficiencies in the pharma industry today are really 20th century issues. In the 21st century, we should be looking to eliminate most of these deficiencies. Currently, we have information silos. Our entire data support system for visual inspection, particulate, and physical defect lifecycle is based on a series of silos, functional silos of information in isolated groups not readily accessible for routine SPC, trending, monitoring, limits evaluation, and deviation investigation. Paper-based information is scattered about also. Information process is stored permanently in paper-based documentation, protocols, reports, data recording sheets, batch records, supplier C of As. This hard copy data is stored in several departments such as incoming materials, production, and QA. Very difficult to really get your hands on this information to do any kind of routine SPC work. Spreadsheets are profuse throughout our organizations, and the data collected from calibration and qualification documentation are routinely transferred to these MS Excel spreadsheets for evaluation and reporting. In many cases, these spreadsheets are not 21 CFR Part 11 compliant. So you can see our data is really all over the place, and even though it works for us, it is not a 21st century solution to the problem. It's also scattered in multiple electronic systems. Even when we do have electronic systems, it's in LIMS, SAP, spreadsheets, often required specialized programming. Often there's no clear roadmap or secure integrated system to allow easily to target any information that's desired for routine root cause investigations and CAPA activities. Custom programming costs and revalidation considerations are prohibitive and stop companies from trying to integrate all of these individual electronic systems. And in this world of big data or Pharma 4.0, in the 21st century, pharmaceutical manufacturers must begin collecting this scattered information and migrate it to a focused digital information application designed to store and evaluate the myriads of data that's hidden in these product defect lifecycle controls. So we have also quality metrics. A big consideration is the key process indicators that exist throughout our product life cycles. This infographic represents the product lifecycle elements of the library, which is a defect informational system, the upstream materials controls, making sure that the upstream materials and APIs come through with controls that are equivalent to the outgoing product quality, visual inspection standards and the calibration of systems and people, qualification of inspectors and vision systems, 100% inspection, visual inspection, and the defect or reject classification of the populations of defects that occur, the AQL, visual inspection process, the forensic particle identification process, and particulate destructive testing. So all of these form the quality metrics that we require to produce and make a batch decision on our products day to day. The first piece of the puzzle is the library of defect definitions for physical container closure defects, particle types, and associated product appearance. That primary use of this library is for creating defect definitions, the name and text definitions of a defect. Criticality designation, which describes the physical defects, container closure, critical, major, and minor categories. It also covers particles and subgroups such as critical A, critical B, major B. So we also store photographic images, macroscopic and microscopic, to display the defects. And many parenteral manufacturers use SOPs to store and display their images in their library. And the poor resolution of these defect images diminishes the effectiveness for training in many cases. So recommended process here is to migrate your library data to digital database applications to store this essential documentation and to facilitate inspector training. Next piece of the puzzle is to work on your upstream control parameters for your defect attributes and metrics. Raw materials prior to bulk formulation. All materials should be liquid and terminally filtered, but in the case where we have API or suspended solids that can't be filtered, they should be pre-tested for particle burden and particle size distribution. Product contact materials, post-washing and processing. For containers, the critical parameters would be physical defects, certainly critical physical defects, particulates, and silicone levels. For elastomeric closures, certainly particulates and silicone levels. Single-use equipment, post-processing, vessel, container, manifold, tubings, connectors, critical physical defects, particulates, and container closure integrity testing. So your metrics focus on upstream controls would be routine databases, not less than yearly particulate trending and limits evaluation for defect attributes that are being monitored. And this would be either from supplier data or in-house data. For current data integrity issues with visual inspection human qualification, all focusing on the standards repository and the qualification training and manual inspector qualification and vision system studies. All visual particulate standards should be documented in a database. A primary concern is the manual qualification data, which is influenced by the bias from the inspectors, and the vision systems we know do not exhibit this bias. Human inspection bias can be introduced by several things. This is not an all-inclusive list, but human readable numbers or code on the defect standards, inspection staff sharing or recognizing specific critical defect containers in a qualification test set, unintentional or unauthorized access to the legend or quality answer key, blank units and defect units that have obvious detectable differences that would allow them to determine which units are easily the rejectable units and which units are the acceptable units. Not strictly following SOP by allowing extended or unlimited inspection time during qualifications. Qualifications of inspectors with USP 790 single container inspection, and then allowing multiple container inspection to occur in routine practice. Obviously, a comparison study would be needed for the multiple container inspection method. Also, test set defect to blank ratios greater than 10% can introduce some form of bias. So whether the ramifications to data integrity on visual inspection standards, the results from inspection issues that could be attributed to inspector bias would be inspectors may not be properly qualified to detect small particles or critical defects. The inspection method, false reject rate, can be underestimated, and this is an economic concern for the corporation. Integrity of true probability of detection, calibration, or baseline data can be questionable. Manual probability of detection data for comparison during validation of alternate inspection methods may not be accurate. And here's a very common example. If a manual visual inspection baseline, if the probability of detection data is falsely inflated by bias, it may be very difficult to show equivalence in an automated vision system. So let's talk about qualification and qualification-related metrics. The evaluation of human inspector training effectiveness prior to actual qualification is a very important factor, but almost no one records this information and documents that to show when an inspector is actually ready for true qualification. So document the actual inspection time and breaks during manual VI inspections. Make sure that is at a worst case scenario, typically end of shift or some other high stress scenario. The inspector or vision system qualification should be include near vision acuity, which requires scheduling and the storage of documentation. Not less than 3 trials for initial qualification, and that requires scheduling and data analysis and documentation. The scheduling and data analysis of requalifications on not less than a yearly basis, documentation and reporting periodic performance checks, tray audits of human inspectors conducted monthly or quarterly, and then the routine daily automated system challenges or pseudo stability checks for automated systems. So your metrics focus in the qualification area is to utilize electronic database management of test standards and calibration POD data, qualification data, and acceptance calculations of human and vision systems. The 100% inspection arena. The metrics involved here is that we must inspect each and every unit for visible particles, critical physical defects, and incorrect product appearance. The inspector should separate rejects at the time of detection into broad categories such as visible particulate, critical, major, minor, and container closure defects. But we should always limit the inspector to those 4 categories. Don't make them do too much. The rejected units hold the information needed to determine if a batch of product falls within the process norm, or if the evaluation requires reinspection to make a product release decision. Action limits based on historical data must be established for total rejects and on primary and subcategories if you have them established. So your metrics focus is to routinely database and trend 100% inspection data and not less than yearly limits evaluation. Database and monitor particle and defect types to support that in-process 100% inspection. Your defect classification, metrics, and monitoring. The 100% inspection process is a cleansing process. It's designed to remove defects from the manufactured product. Initially, this is done for visible particles, critical, major, and minor categories. Further classification, sometimes called reclassification, is performed after the initial separation into more specific subcategories. Particulate characterization at a high level is performed by separating into basic intrinsic particle groups like glass-like, stainless steel-like, elastomer-like, polymer-like, fiber-like, or other undetermined particles in situ. This same concept is applied also to physical defects of the container closure system. Initially, for baseline, conduct a full classification of all the rejects, and then later make a justification for statistical fractional classification. Your metrics focus here is to routinely database and trend 100% inspection reject population data, and if possible, establish action or alert levels for particles and physical defects. Next piece of the puzzle is the AQL inspection. The AQL inspection is the statistical samples that are re-inspected by the identical 100% inspection method to determine the effectiveness of that initial 100% inspection. And we look at the defect frequencies that remain undetected in that good fraction. Remember that USP 1790 recommends various categories of criticality and AQL limit ranges. And for intrinsic particles, we have to realize that less than or equal to 0.65% AQL is really in the major defect range. Critical, major, and minor ranges are also listed in USP. These rejected units hold the information needed to determine if a batch of product falls within the process norm, or if further investigation or reinspection is needed to make a product release decision. So metrics focus for AQL is the database and trend AQL inspection data and not less than yearly limits evaluation. database and monitor your particle type populations coming out of AQL inspection. The importance of AQL particle identification monitoring is that these particle types that you find during an AQL inspection are resident in the good product fraction. That means this is in the product that we are intending to put on the market. The identification of different particle defect types facilitates deviation investigations. The characterization is typically done in a level 1, level 2, level 3 methodology. Allows us to get the full information necessary to characterize these defects in the forensic laboratory. So this forensic laboratory or particle identification documentation is also very much useful for updating your product library with new particle types as they are recovered. So a metrics focus for the forensic data is to database and monitor particle types, particulate population trending, and evaluation of intrinsic, extrinsic, inherent, and other particle types is essential, and to look at sensitive particle types such as foreign particle types that are extrinsic and glass and metal particle types. Destructive particle testing is the last piece of the puzzle. Subvisible particles going by USP subvisible chapters or the ophthalmics chapter, and to realize that we don't want to just report meets USP limits. These limits are very large, they're very wide, don't represent the true running average of most of our products. So we must establish action levels based on historical data, and our metrics focus needs to be on particles greater than 10, 25, and 50 microns with routine testing and not less than yearly particulate trending and limits evaluation. For visible particulates in difficult to inspect parenterals, we are following the guidance of USP 790 and 1790 for supplemental testing to 100% inspection. This is similar to the 0.65% AQL for essentially free, and identification of recovered particulates is essential. The optional information is to more stringently look at other specific types if required, such as extrinsic, glass, and metal. So this is the particulate destructive testing for both subvisible and visible, and your metric focus here is particulates greater than 100 microns, routine testing for SPC, and not less than yearly particulate trending and limits evaluation. So also, it's important to database and monitor these particle type populations. So the conclusions based on visual inspection data integrity is effective VI calibration, qualification, and or validation is essential and must be taken very seriously to maintain data integrity. The VI and supervisory group must assure that all units are blinded in a test set and should have non-human readable code. Test set defect standards during use must be procedurally secured and documented in a clear chain of custody. Protect your data integrity by maintaining compliance to inspection pacing, timing, and unbiased blinded test standards for qualification of humans. Consider introducing more automated vision systems with more sensitivity and consistency than humans. They don't have bias. Move towards paperless digital databases that are 21 CFR Part 11 compliant for real-time recording, databasing, and trending. And our conclusions for the metrics approaches would be to utilize this concept of this infographic, this life cycle infographic. The quality metrics and key process indicators exist throughout that product defect control life cycle. Utilize the data from each functional area upstream in process, finished product, to enhance the controls and limits through the trending and monitoring process. Plan to begin the deliberate migration from paper-based and spreadsheet evaluation to 21 CFR Part 11 compliant databases for digital management of your data. And this integrated data is essential for you to keep control in the 21st century. So 21st century digital solutions are being developed for connecting the big data, metrics, trending, monitoring, and data integrity controls associated with the visual inspection, particulate, and physical defects lifecycle So select your appropriate application solutions and tools, plan your transition in a phased approach, and I thank you for your attention.